Today we are going to discuss inhibition of prostaglandin degrading enzyme 15-PGDH and how this potentiates tissue regeneration. This study was published in 2015. This research focused on finding a new method to improve hematopoietic stem cell transplantation in people. This is important due to the current side effects associated with tissue transplant, including rejection of tissue, infection, and internal bleeding. Specifically, this can improve treatment of ulcerative colitis and hepatic carcinoma. In order to understand where we're going, we should know where we've been. First, they relied upon a study which focused on umbilical cord blood to augment stem cell grafts. UBC tend to have reduced progenitor cells, so they tried to improve engraftment capacity with cytokines, growth factors, and other molecular targets ex vivo. Currently, there isn't enough data in vivo for this to be a target of clinical trials. Second, they referenced a study that stimulated expansion ex vivo of human cord blood cells, which could reconstruct human hepatoesis for 6 months in immunocompromised mice. The preferred molecule they identified was UM171 for stem cell transplantation and gene therapy. Third, they referred to a study which also utilized UBC. They demonstrated 1616 dimethyl prostaglandin E2, or DMPGE2, treated UBC improved transplantation and is currently taking place in clinical trials. Keep this in mind as DMPGE2 will be coming up again later. Whew! Now that we have seen what has been done, let's focus on what this could directly improve. Ulcerative colitis is an inflammatory disease with symptoms like GI bleeds and hematochezia, which is basically bloody diarrhea. They usually treat this disease with immunosuppression, which has its benefits but doesn't restore the damaged tissue. Next is hepatocellular carcinoma. This disease is usually caused by chronic hepatitis, and symptoms include jaundice, which is yellowing of the skin, abdominal pain, and weight loss. Treatments include surgery, which this study touches on, or chemotherapy, which again provide benefits but does not allow the organ to regain function. As you see, tissue transplantation could significantly improve the lives of patients with ulcerative colitis or HCC. Now let's discuss our molecule of interest, prostaglandin PGE2. Prostaglandin PGE2 is produced by COX-1 or COX-2 via prostaglandin E synthase. Prostaglandin PGE2 aids in WNT signaling, which more or less performs maintenance of stem cells. Research already shows that prostaglandin PGE2 in conjunction with DMPGE2, as we recall from the research discussed earlier, improves hematopoietic cell numbers in mice and zebrafish. As previously stated, DMPGE2 is already being used in clinical trials, so today we are determining if prostaglandin PGE2 could be a better target. So, how do we increase concentration of prostaglandin PGE2? Either by increasing synthesis in the cells or inhibiting the degrading enzyme, which is 15-hydroxyprostaglandin dehydrogenase. Today we will focus on silencing 15-PGDH. First, does 15-PGDH really regulate PGE2 in vivo? They measured PGE2 in 4 different cell types, as you can see on the right, in wild type versus knockout PGDH cells. As you can see, there tends to be double PGE in wild type cells versus the knockout 15-PGDH cells. Therefore, targeting 15-PGDH could increase PGE2 and lead to better reception of HSC transplantation. Next, they determined if 15-PGDH really inhibits the positive effects of PGE2. Again, they compared 15-PGDH mice to wild-type SW033291 treated mice, since this inhibits 15-PGDH activity. There was a 43% increase in neutrophil counts and a 39% increase in specialized bone marrow counts in knockout mice versus the wild type. The improvement continued 3 days after treatment, as you can see on the slide. Following, they performed assays of bone marrow to determine the difference between knockout 15-PGDH and SW033291 treated cells. Results, which are given on the right, demonstrated 55% increase in hematopoietic colonies after plating, and then a further 50% increase in the SW033291 cells only. The next step focused on SW033291 effects on gene expression. They collected bone marrow cells from control mice and SW033291 treated cells. Results showed an increase in PGE2 levels, and that this specifically targets EP2 and EP4 cells to induce CXCL12 and SCF responses, which are significant cytokines for hematopoietic stem cell homing and maintenance. Next, they wanted to determine if SW033291 improved transplantation in mice. That identified a 2 to 3-fold increase in the number of donor cells in comparison to the control. In addition, they noticed that this could be blocked with an inhibitor of PGE2 generation, an inhibitor of PGE signaling, or an agonist of CXL12. With these experiments, they demonstrated 15-PGDH inhibition leads to an increase in bone marrow PGE2 and increases CXCL12 and SCF. Next, they took irradiated mice infused vehicle-controlled donor cells and SW033291 treated donor cells. As you can see on the graph, control mice died in 6 to 14 days, whereas SW033291 treated mice survived over 30 days. Following, they looked at hematopoietic recovery with SW033291. Mice were treated with 500,000 control bone marrow cells and SW033291 treated bone marrow cells. Results showed most control and SW033291 survived 30 days. However, SW033291 had a faster recovery of neutrophils, red blood cells, and platelets. With these experiments, they demonstrated that 15-PGDH generates increased PGE2 and avoids extremes of concentration when performed exogenously. Alright, now let's revisit the ulcerative colitis problem. Mice were treated for 7 days with dextrans sodium sulfate to cause colonic ulcerations. Mice were then treated with SW033291 and a 15-PGDH knockout in comparison to vehicle controls. As you can see by the graphs on the right, there were 70% less colon ulcers 87% less area of ulcerative colon mucosa, reduced mucosal inflammation, reduced symptoms, and reduced colitis-associated inflammatory cytokines in comparison to the control. Next, chimeric mice received transplants from tissue with knockout 15-PGDH and wild-type donors. Both showed identical susceptibility to DSS-induced colitis. This indicates colitis protection is due to inhibition of 15-PGDH in colonocytes rather than inhibition of 15-PGDH in bone marrow-derived cells. Following, they looked at SW033291 effects on proliferation in colon crypts. 7 days after DSS treatment, wild-type cells received addition of vehicle control SW033291 or 15-PGDH knockout cell types. The results show a 2.5-fold increase in SW033291 mice and a 2.4-fold increase in 15-PGDH knockout mice, as demonstrated on the right. These experiments demonstrate 15-PGDH inhibition leads to protection after treatment with DSS. Now, let's revisit HCC. 15-PGDH knockout in SW033291 treated mice showed increased rate and amount of liver regeneration in comparison to the vehicle control. Specifically, knockout mice had improved weight by 28% by day 3 and 40% improved weight by day 4. In comparison, SW033291 showed improved weight by 77%. Okay, we've made it through. Let's review what we found. The 15-PGDH enzyme negatively regulates tissue regeneration and repair in bone marrow, colon, and liver. PGE2 and DMPGE2 improve tissue regeneration when used to treat cells in culture and when administered in animals. 15-PGDH inhibitors like SW033291 can improve HSC transplantation by increasing safety and reducing required time of hospitalization due to side effects. And finally, 15-PGDH inhibitors like SW033291 could be a target of treatment for ulcerative colitis by improving mucosal healing. Now it's your turn. What do you think?